I'm Thomas.
I'm Bradford.
And I'm Jonathan.
And we're students at Brigham Young University in the Bio department. And today we are going to report on our findings of the bacteriophage Saratos. It's a bacteriophage that was isolated last semester and annotated this semester. And today we're going to present the information. So Saratos is an EA2 subcluster bacteriophage that contains 40,351 base pairs, 63 ORFs, or open reading frames, 29 forward and 34 reverse, and has significant coding potential. The phage was initially isolated in 2020 from a compost sample, underwent several purifications until prepared into a high titer lysate, and then the genome was sequenced and annotated. Saratos is in close relation to Ellery, Microbacterium, Glamour, and Colocorda, all of which are bacteriophages in the EA2 subcluster. With 7 comparatively rare ORFs found in less than 10 other bacteriophages, our data show that Saratos has a unique genome.
These are some of the methods that we used to annotate Saratos's genome. Right here is a program called DNA Master. We supply DNA Master with the amino acid sequence, and it does a full auto annotation of the genome. There are also tools inside DNA Master that allow us to look at the open reading frames and determine longer and shorter coding potentials. They also show us all 63 genes. We also use programs such as Famirator to compare Saratos to the other bacteriophages in the EA2 cluster. Famirator also allows us to look at each individual gene. Glimmer and GeneMark provide us with a potential start and stop codon from the protein sequence, which is used inside DNA Master. NCBI BLAST and HHPRED give us the protein length, coding potential, protein function, and percent identity match compared to other bacteriophages. Let's take a look at gene 14. This heat map was created using MAFFT, or MAFT, which constructed an alignment of the different genomes and calculated percent identity. MAFT is a multiple sequence alignment program. It offers a range of multiple alignment methods. We uploaded the matrix provided by MAFT to Morpheus to produce a heat map. Morpheus is a virtual matrix visualization program that allows you to view data as a heat map. These two programs are important in the comparison of gene 14. As we were analyzing Famirator's call for gene 14, it led us to believe that the nucleotide sequence is shorter than that of the subcluster, about 155 base pairs long. Further analyzation of gene 14 led us to discover that it is 561 base pairs long, opposed to the 346 average nucleotide base pairs of the corresponding genes of the bacteriophages in the subcluster. In other words, gene 14 was longer than anticipated. Since gene 14 is so much longer than the other genes in the subcluster, we compared them to find identity match to see how important this difference was. The data show that gene 14 in Saratos is significantly different from the corresponding genes of M cubed, Glamour, Illyri, Colocorda, Endometas, Finney, and Sansa.
We also found that Saratos has an average nucleotide identity match of 98.24% with other bacteriophages belonging to the EA2 subcluster, such as Illyri, Glamour, Colocorda, and M cubed. This can be seen in the graphical and numerical representations below, with the largest difference being a difference of 3.25% between Saratos and M cubed. Saratos has 7 ORFs, or open reading frames, which belong to families containing less than 10 members. If the true mean number of members in a family found in any of the EA subclusters is 104.19, the probability of a fam having less than 10 members is 2.11%. Statistically, any probability less than 5% is considered significant, meaning that fams with any less than 19 members would be considered rare. 15 of the 63, or 24% of the ORFs, or open reading frames, found in the Saratos genome meet this criteria. This chart represents a statistical analysis of a simple random sample of fams found in bacteriophages belonging to the EA subclusters, while highlighting the 2.11% of fams which have less than 10 members. In conclusion, our results show compelling evidence that Saratos is closely related to other EA2 subcluster bacteriophages. It also shows that it contains 7 comparatively rare ORFs, or open reading frames, as well as a unique sequence for gene 14. Saratos is a novel genome, and our analysis of it contributes to the information regarding EA2 subcluster bacteriophages. We would like to acknowledge Brigham Young University and the microbiome department, as well as C-phages and the University of Pittsburgh for their contribution to our findings today.